webinar. But anyway, here I'm presenting an elemental atlas of your plate, ICP MS-based ionomic analysis of 500 foods at CSU. As Corey mentioned, I am the person who runs the ICP MS platform in ARC, and I also am a research scientist at Jessica Prenny's lab, managing her lab and all the instrumentation there. But let's get started. Food is essential to human life. Food is a biological necessity. We need it for survival, but food is more than just a means of survival. It is tied to cultural identity and heritage. The passage of recipes, ingredients, cooking methods helps preserve one's heritage. Meals bring people together. Think of family dinners during the holidays or celebrations. They help foster relationships and communications. Food is a means of learning about different cultures, histories, and geographies. I know anytime I travel around the world, I make it a point to schedule a food tour to really be able to understand the area that I'm visiting. And food also evokes joy and nostalgia. This past holiday season, helping my mother-in-law make fresh pasta and all the dishes that are traditional for an Italian Christmas dinner really brought everyone, all of us together. And so, as I mentioned, food really is something that helps us communicate and spend time together. But with that being said, there are knowledge gaps. There are knowledge gaps in food composition. Here on the left, you can see what you typically see of a nutrition facts label of an apple. You have calories that are present, but what really is food? These nutrition facts contain amount of a few elements found in foods such as calcium, iron, sodium, and potassium. But what drives these changes in these foods, besides different things? So we can observe this apple in the orchard and see what from the moment the tree blooms until the apple reaches our plate, its chemical composition undergoes continuous changes. Once the apple reaches our plates, this can be related to these changes can be related to both abiotic and biotic factors such as pests or environmental factors, drought, pathogens, management practices, transportation, and storage. And in each one of these steps, different processes can occur for which, the elemental profile or the biochemical profile of our foods changes. And so greater understanding regarding food composition, including the diversity of elements and their quantities, has the potential to empower stakeholders throughout the food system to make evidence-based decisions to better address both the health of people and the planet. And so in order to kind of start looking at that is, we are involved in a project, the Periodic Table of Food Initiative, and that is and the mission of the PTFI is to build a global ecosystem and provide data, tools, and training to enable us to catalog the biomolecular composition of the world's food supply. So collectively, these tools will enable data-driven solutions to support human and planetary health. So how are we trying to do this? So the PTFI Toolbox for Advanced Knowledge has built data, so standardized analytical methods that we're able to use to generate comparable data across all our collaborators and researchers across the world. A database and interface where all this data can be found, and it could be comparable data for researchers that then they could use and compare with the results that they've had, as well as training. So Food EDU, we're developing educational offerings for diverse learners around the world to strengthen the capacity of scientists, educators, and policymakers to both generate and utilize this next generation of food composition data. And so who is a part of all this? So we're building an ecosystem worldwide with partners all across the world in every continent except Antarctica. And we're building this global ecosystem to help build local capacity and empower communities to explore their native food sources. This will contribute to a global understanding of the composition and diversity of our foods, thus making this a global effort. And so then all these labs, not only are we sending these protocols, the standardized protocols. We're training, and everyone can then upload and leave their data at this public repository so that we can start looking at and answering different questions that are necessary. As we discussed, food is remarkably complex and diverse. This image showcases a range of protein sources, vegetable grains, and dairy products. And so to truly understand the elemental composition of food, several challenges must first be addressed and overcome, especially since food matrix is definitely one of the more complex matrices that I have worked with. So some of the things that we have to keep in mind is that elements vary within food. So we can look at this purple potato, Purple Majesty, that was actually developed here in the Department of Horticulture in our extension and the potato breeding program. And we can look at this potato, and we could see that, and we can look at how much arsenic is in this potato. And you can look at, pretend this pink little dot represents 1x time of arsenic found in this purple potato. But then we also have to take into account that we also want to study how much iron might be in this potato. And that tends to be 80 times more concentrated than the amount of arsenic that is in our potato. And furthermore, if we want to look at macronutrients at the same time, and we look at something like phosphorus, this tends to be 490 times more concentrated than arsenic in our potato that we're studying. So then we have to figure out a way to be able to not only understand the concentration of a single element, but of all the elements that we're interested in, and know that these vary within a food. So when looking at a single food, we can look at that. But another issue is that if we want to look at, in this case, 500 foods, we also know that elements vary across foods. So here we have a sample of corn that the little pink circle, you could make it represent 1x time concentration of calcium. We also want to be able to also measure the amount of calcium in whole milk, which tends to be 14 times more concentrated than that we find in corn. We also want that of bok choy, a leafy green, and that is 19 times more concentrated than the calcium found in corn. And furthermore, in black beans, which is 5 times more concentrated in corn. And so as you can see, we might have a panel of different elements that we want to analyze, but we also have to take into account the fact that we want to analyze different elements in one food, and they vary in concentration, and that even if we want to measure one element across different foods, that varies, that those vary also in concentration. So, and why is this important? Element imbalances result in health issues. If you have too much of zinc or iron, that you can have different issues. If you have too little of zinc or iron, there are other elements. So everything needs to be just right and in balance so that we can increase human health and productivity. This is why we need to understand both quality and quantity of the elements in your food and what that really means when everything is combined. So with all that background, we can now go in and talk about looking at the first 500 foods that we looked at. So we compiled a list of 500 foods. 357 of these were plants, 59 animal products, 32 aquatic foods, 8 fungi and bacteria, and 44 complex foods that include highly processed foods such as fast food, burgers, and chocolate. And these complex foods also, and all these foods represent 69 different families across the phylogenetic genetic tree. So we're adding some diversity, and we're also understanding that individual foods are different. So a green apple does not necessarily have the same elements or makeup as that of a red apple. The food components, we eat beets and the beet and the beet greens. So the leafy part of the beet greens would have a different elemental composition than that of the actual beet. And even preparing, when we cook a sample, whether raw or cooked, the elemental profile can change as well and can be distinct. Additionally, within the complex food, we have chef curated dishes that we obtained from a chef, and we know exactly every ingredient and the amounts that were added to each food. So now we can add some complexity to our analysis and understand not only how a single food can affect something, but when we're combining into a recipe different foods, how does that elemental composition might change or how it interacts with one another. So in order to do that, like I said, we made what is called the PTFI method for element analysis. So this includes 26 elements that are perceived to be important for human and plant health. So we have macro elements such as sodium, magnesium, potassium, calcium, phosphorus, and sulfur. We have micro elements that are boron, manganese, iron, copper, zinc, selenium, and molybdenum. And we have trace elements such as lithium, beryllium, vanadium, chromium, cobalt, nickel, aluminum, arsenic, strontium, cadmium, barium, tungsten, and lead. And so in here, we're looking at not only elements that are important for human health, but also that cause illnesses such as these heavy metals, lead, arsenic, chromium, and such, and cadmium. Going further, we have standardized food procurement and processing as all foods are lyophilized and then homogenized and aliquoted for analysis. Standardization of food procurement and processing is essential, but also obtaining metadata on the range of factors to enable data comparisons and integration. Let's consider one of these apples. So in here, we want to record not only where it was grown, because we know soil and growing, this apple or this food influences the elemental composition. We also want to know what management practices were used, such as organic or conventional methods, where it was obtained, whether from a supermarket, farmer's market, or directly from an orchard. And what type of preparation it underwent prior to analysis. All these things will then give us a clearer picture of how knowing all these parts of how the food was processed and stuff like that will give us a better idea of how the differences in elemental profile might have been affected by some of these things. That also helps us, getting all this data also helps us in the public database being able to look at results from other researchers and seeing how we can compare the results based on the either experimental design or where these foods were grown and processed. So after procurement, then we perform microwave digestion. We weigh out 200 milligrams of sample. We add 10 milliliters of doubly distilled nitric acid spiked with our internal standard indium. Then samples are digested using the PerkinElmer MPS 320. Once the samples have been digested, they are transferred and diluted to 25 mils using type 1 water. Then 1 ml of solution is taken and diluted further with type 1 water to 15 mils to a final concentration of about 15 parts per billion of indium and about 2% volume to volume of nitric acid. So this ends up helping with all the 26 different elements across different ranges, macro all the way to ultra trace elements. This dilution factor will help us be able to measure all those 26 elements from one sample alone. We also then, in order to do quality control, we make a pooled QC where we take equal volumes of each food sample digestion and create a pooled QC, which will be analyzed after every 10 samples. So this is quality control that can be as closely matrix matched to the different food samples that we have, and we can understand how this matrix and how the analysis changes over time due to various different constraints. Then samples are then analyzed using a PerkinElmer Nexion 2000, where we analyze the 26 elements as well as internal standards. So we have the internal standard indium that was added to control for the every time from sampling and sample processing and microwave digestion all the way to analysis. But we also infuse 3 other elements: rhodium, iridium, and the lowest isotope of lithium, lithium-6, to be able to for quality control and be able to understand any drift or any issues that are happening. We also use the dynamic reaction chamber using ammonia and oxygen gas, and we also measure multiple isotopes to avoid interferences. So let me give you a little example on why we do that. So with the acquisition, as I mentioned, we measure the internal standards, indium that's in the sample, lithium, rhodium, and iridium that are also then infused into the system to be able to understand, as I mentioned previously, what is going on. We also use the dynamic reaction chamber because food matrices are so complex. So then we use the carrier gas. We use the gas oxygen, used for analysis of selenium, sulfur, arsenic, and cadmium to overcome some interferences. And then we use also the ammonia gas to overcome interferences for aluminum, vanadium, chromium, manganese, iron, cobalt, nickel, copper, zinc, strontium, molybdenum, cadmium, barium, and iron, as an example. So why do we need to really do this? So we can look at iron here. Our most abundant element of iron, or isotope of iron, is iron 56. But iron 56 has large interferences with argon oxide, and argon is what we use to make the plasma. So our ICP MS is filled with argon gas there, and the oxygen is just in nature. So then in order to avoid these interferences, not only do we use the dynamic reaction chamber with ammonia gas, but we also measure iron at lower stable isotopes in terms of abundance, iron 54 and iron 57 as well. So we measure both in standard mode as well as in ammonia gas. We do similar things with elements such as sulfur, argon, aluminum, vanadium, and selenium, and arsenic, to name a few. And so this is done because we don't fully understand everything that is in our food matrices, and we want to make sure that we can capture any potential interferences and be able to control them and not have to reanalyze our samples. As I mentioned before, we measure a lot of macro elements that are in high abundance and concentration in our samples. And due to their concentration, they can sometimes overwhelm or shut down the detector. As here you can see our cartoon the person in our cartoon is our detector, and we can see potassium ions just flying in and hitting this detector and not knowing what's going on. And so what we can do is adjust the bandpass parameter, or the RPA, to attenuate the signal of macro elements. So then less potassium ions are present. So you could think of this as a, so I might be dating myself, as a shutter or the aperture of a film camera, where the higher the diameter of the opening, the more light comes in. The smaller that diameter of the shutter, less light comes in. And so then you could adjust that to be able to overcome some of these very concentrated elements while still being able to capture the really low concentrated elements that we're also interested in. So once we have ICP MS data acquisition, the next thing we have to do is data processing, which occurs using internal standards for normalization and pooled QCs for drift correction. Then we could do quantitative analysis and multivariate and univariate analysis to be able to start understanding and quantifying what is going on in our samples. And so now, with all that background and how this process is done, let's look at our results. So here, using hierarchical clustering analysis, the elemental composition of the foods divided the foods into 5 groups. So group 1 has 55 foods and mainly comprises of leafy greens such as spinach and bok choy, and some seaweed. Group 2 includes 143 foods and comprised of non-dairy milk products such as oat and almond milk, prepared foods such as highly processed foods, and insects. Our group 3 contains 67 foods and comprised mainly of animal products such as cheese, beef, poultry, and fish. Our group 4 included only 3 foods consisting of fat sources, sunflower oil and butter derived from both goat and cow milk. And that was really interesting and kind of a really neat thing to see, because these are the only 3 foods that out of all these 500 foods that that we selected that are mainly these fat sources. And so that was pretty cool to see in terms of that separation in elemental composition. And group 5 included 162 foods and comprised of nuts, grains, and legumes. And so it's really hard to really analyze all these 500 diverse foods, but we can start seeing that some of these foods, based on their full elemental composition of 26 elements, start separating and being distinguished from each other in trends. So now when we're looking at the foods that the elemental profile as a whole, we can also look at specific elements and start identifying foods with high that are either high or low in essential elements. For example, calcium is very necessary for our bones and our teeth and our health. And then we could try using in this plot, every dot represents a food sample, and then every food was divided into the 5 groups that were represented by the hierarchical clustering. And then these are all in concentrations of grams per 100 grams of dry food. And so then we could start looking at that non-dairy milk, such as various sources of almond milk and organic almond milk, contribute to high calcium. So these almond milk, we can look and see if they were fortified with calcium or if there are other things that are happening. We also see that there's a edible weed, the Canada thistle, has high calcium as well as an organic red kale. We can also start identifying foods that can help alleviate nutrient deficiency. So again, in 2021, over 1.9 million people are living with anemia. Thus, possibly finding foods high in iron can help us begin to mediate this. While we know bioavailability of iron is still most important to increase iron in the body, this can still help us start looking at potential candidates and seeing how we can start looking at different foods that can help alleviate that. So for example, if we look at group 1, that is, it has really high levels of iron. Canada thistle again, that we were looking at, the edible weed has high levels of iron. Now, the biggest thing to note is that group 1 is under the y-axis on the left. So you see that concentration is much higher than that of groups 2 to 5. And so it'd be interesting to kind of see, well, we know for the most part, heme iron is more bioavailable. Well, how can we do something about these leafy greens and Canada thistle that might have also higher iron levels? We also see notably beef liver is high, that's in group 3, but also that black olives seem to contain a lot of iron as well. We also identify foods that can help alleviate nutrient deficiency with looking at zinc again. Similarly, zinc, these foods, we can see that that there's specific foods high in zinc that could potentially help with this. For example, cricket powder has the highest amount of zinc in group 2, and cowpeas from group 4 also have high levels of zinc. Again, this is a simplistic view of the problem. We can start identifying candidates that can help ameliorate these issues or that need further exploration. Now, with looking at things to help nutrient deficiency, we also want to be able to identify foods that have high toxic elements. So, for example, we looked at the distribution of total arsenic across the 500 foods, we see that seafoods and seaweed have high concentrations of total organic acid, arsenic. But again, this is not the full picture, as all forms of arsenic are not toxic, and thus these foods high in total arsenic can then undergo further testing by performing speciation analysis to determine if we need to worry about these high levels of total arsenic. So studies, we have started identifying foods that are high in arsenic and starting to perform speciation analysis to determine, do we really need to be worried about that, or are most of these arsenic compounds or species organic arsenic forms where we have less to worry about. So what are we doing with all of this? We're building a roadmap and identifying foods that can help mitigate mineral nutrient deficiency and those that need further evaluation for toxic element content. So we're looking at 500 foods here, identifying what's present, what the concentration, the levels are, how they are dispersed across all the foods, and then we can start asking, having this data, we can start asking more relevant questions with regard to, as I said, how can we help increase iron bioavailability by starting to look at these foods that are high in iron. So as I mentioned before, the PTFI is building a toolbox to advance food knowledge. We have heavily talked about the data collection, but we also have like the database where all this data can be found freely available, and which is Marker Lab, which is an interactive database on the food. And this, you create an account and you're able to start seeing all the different elemental components in food, but as well as it's a repository where we've looked at different components of the food, such as metabolomics, lipidomics. We have some protein data in there, some FAMES analysis, and proximate analysis, and we can start building and understanding all the different components in food that are present. We also, with respect to the trainings, we have that food EDU training that is also a big focus of PTFI through the American Heart Association. And then I have helped develop an Ionomics tutorial to be able to look at all the different how to use ICP MS or to understand the elemental profile of different foods. Additionally, in the ARC where we're doing, while I focus most of this talk on food, it doesn't matter what kind of sample we have and what elements you're interested in. We are definitely able to analyze and look at different sample types to be able to answer the questions that you need. So with that, I want to say thank you, and to all the people that have guided and helped me in this, and I guess if you have any other questions or any questions.
Thank you, Jackie. We'll open it up for questions. If you happen to not be at a microphone, feel free to text in the chat, and we can ask verbally for you. So I have a question, maybe to kick things off. It looks like there's a pretty distinct pattern in sort of food class, beans and grains, et cetera, et cetera. How much does your data tell you about how some of these elements covary? Like I would guess, for example, that a lot of the divalent cations may covary just because the biological properties or the biological reactions that contribute to their accumulation in the food has a difficult time distinguishing between them, but maybe that's not true. I was curious to know if you looked at any of those sorts of patterns.
we're still exploring the data, and so those are definitely things that we want to look at. But we have seen that at least, for example, location where things were grown and the soil that was grown really has a pretty significant effect on the elemental profile, much less so than the genetics. So that can help us start understanding, you know, the growing location and even management practices, how we can influence those to potentially reduce toxic elements or increase elements that are important for our health.
Other questions? I do have one more. I remember the arsenic slide, you had lobster and shrimp as having relatively high levels. Given what you just commented on about the source growing location, is there a lot of arsenic in seawater? Or
There are pockets of that in terms of but the nice thing about the seafood and what we've like I said, we have done some speciation on this and a lot of it is organic forms of that and we're also doing a more extensive review of seaweed that we also saw had really high levels of arsenic and seaweed right now. You know, it's very popular for people to eat, you know, dry seaweed, in sushi and even, we see in grocery stores now and in health stores, to eat dry seaweed and looking at how these high levels are present. And also we collaborating with a group in one of the collaborators in Thailand, who is part of PTFI, as well as with some farmers in Canada that collected seaweed for us of different forms and looking at not only that we see high arsenic in there, but also now we're going to do speciation and seeing, okay, do we need to be worried about these arsenic, this arsenic that we're ingesting, and does it differ based on, the region, Canada, I guess, and Thailand in this case that they were sourced.
other questions? Okay. Well, thank you, Jackie. Thanks for everybody for attending. We will be posting some